Welcome to today's educational webinar, Gene Therapy 101. This webinar is being recorded and you are participating in listen-only mode. If you wish to ask any questions at any time, please do so in the Q&A box. We will allow time for questions at the end. My name is Katie Sale, Executive Director of the American Brain Coalition. The ABC is proud to sponsor today's webinar, along with the American Society of Gene and Cell Therapy and MTM-CNM Family Connection. I first learned about gene therapy during this year's NINDS nonprofit forum. I hadn't realized that gene therapy has been studied for more than 40 years and can help stop or slow the effects of the most basic level of the human body, our genes. During the nonprofit forum, I recognized that the ABC membership could greatly benefit from an overview of gene therapy. Allison Kajowski at the American Society of Gene and Cell Therapy was instrumental in helping to organize today's webinar, and I am thankful for all of her hard work and support. I now invite you to learn the basics of gene therapy and how it works, as well as hear from patient advocates who are supporting families interested in gene therapy to help treat their disease. Our speakers include Nancy Kuntz, MD, attending neurologist, Anne and Robert H. Lurie Children's Hospital of Chicago, and Erin Ward, President, and Marie Wood, Vice President, MTM-CNM Family Connection. We will first hear from Dr. Kuntz, who is representing the American Society of Gene and Cell Therapy. Dr. Kuntz is the attending physician, Division of Neurology, Medical Director, Maza Foundation Neuromuscular Program, Director, Muscular Dystrophy Association Clinic, and Professor of Pediatrics and Neurology at Northwestern University Feinberg School of Medicine at Lurie Children's Hospital of Chicago. And that is a mouthful. Yeah. Dr. Kuntz was the expert voice in the American Society of Gene and Cell Therapy patient-friendly video explaining gene therapy for X-linked myotubular myopathy. Please join me in welcoming Dr. Kuntz.
Thank you very much. I can assure everybody that gene therapy was not something that was anticipated within our years of practice as my class of people went through medical school a number of years ago. So it is with great excitement and a need to really continuously update my knowledge base and my skills that I approach gene therapy. I'll walk you through in just a few slides an overview of what is being introduced. But one of the most important things is to realize that gene therapy is not a single type of intervention. And we'll talk a little bit more about the different pieces of it as you go. But I thought we'd start by just considering that the building blocks of the body are the cells. We have trillions of cells in our body, and that's what you see here on the left of the slide. The oval circle in the middle is meant to represent the nucleus, and that nucleus within every cell has, amongst other things, the genetic material, or the chromosomes, that provide the guidance for how to build proteins that are both structural as well as functional, meaning things that would facilitate or make chemical reactions occur and other kind of transformations occur within the body. You can see that the chromosomes look like little X's for the most part, although you'll see in the next slide they're shaped very differently. But when you take each of the limbs of the chromosome apart, you see it is this helix, is what people call it, a triple helix of twisted DNA. Maybe we can look at the next slide. This is just to remind us that in every cell, except for the germline cells, meaning the sperm and the egg, in all other cells, women and all females have 46, including 2 X chromosomes, and then all males have 46 with 1 X and 1 Y. And so you can see that each of the different chromosomes look very different. Some have even lengths between the joining in the middle. Others of them are kind of stubby on the top and long on the bottom. They have different widths, and the different intensity of black has to do with how tightly coiled and how tightly packed different parts of the chromosomes are. Now, it's only been within, I think, this generation that we've had a good sense that all of the disorders that are genetic occur because of a command and control change or a mutation in one of the genes that's just in one spot on all of this complement of chromosomes. Next slide. This is again just to remind us that those chromosomes are made of a tightly coiled DNA or deoxyribonucleic acid, but easier to say DNA, and that is transcribed into mRNA, messenger RNA, and there are several steps there actually, which is just the ribonucleic acid, and then that actually commands production of proteins. Now the proteins again can be used for building. They can be used for synthesizing things or breaking them down or other kind of chemical reactions. Next slide. And this is basically talking about or giving us a sense of what happens if we have an alteration or mutation in one of our genes. There are so many genes, and this whole process in the human body is so complicated that all people have mutations in their genes. Now, sometimes this is something very mild that we can overcome. For example, if you have a mutation in the gene that produces lactose, which is the enzyme that helps you break down, or lactase, the enzyme that helps you break down lactose, the protein in milk, then you're, lactose intolerant. And at least these days, you can have a different source of calcium. So some mutations are not serious or fatal. Other times, that same kind of mutation can occur in a gene that's very critical for functioning in the human body. In the ways that mutations can, or it can produce an abnormal protein that just doesn't function or accumulates in a way that that creates abnormalities. Next slide. So gene therapy is really a group of types of therapy, and the unifying part of gene therapy is that in some way there's either introduction, removal, or change to genetic material within the patient's cells. Next slide. To give you a sense of the types of gene therapy that are theoretically possible, and not all of these are being done at this time. If you go to the left of the slide, you'll see gene replacement. Now, in this circumstance, a person has a mutant gene that is not functioning, and therefore they have a disorder or a disease. And what happens is that scientists and a medical team work together to create a synthetic, perfect functional gene, and in some manner replace that into the human body. And then that would then correct the function. This is the cornerstone of many of the types of gene therapy that are being trialed at this time. Gene addition is slightly different. In this case, there's a disease that's occurring because of, again, genetic factors or things in the environment. But what is being suggested is that scientists would create a therapeutic gene that would produce some function. and therefore alleviate or improve the functioning in somebody. Here, it's not a matter of replacing an abnormal gene, but adding in a new gene. The third panel talks about gene knockout or knockdown. A few slides ago, we talked about how sometimes mutant genes can produce abnormal proteins that can accumulate and actually cause a misfunctioning of the cell or something unwanted to happen in cellular function. And in this circumstance, gene therapy would be brought in to introduce some genetic material that would in some way knock down, silence, or kind of take out the abnormal gene so that the abnormal protein would no longer accumulate. And then the final panel on the right is something called gene editing. People have heard of this many times as CRISPR technology. And here, what's happening is that material is, and frequently the techniques that are being trialed are taking bacterial genes that actually cause their mischief by altering or changing genetic material, and taking those in a controlled way and putting them in to a situation where they will be commanded to fix or change the mutation in the existing gene, so that rather than having abnormal function, you have normal function. CRISPR technology is not yet being introduced. And again, there's a lot of science going on around the gene knockout and knockdown, and the addition that those, in my, to the best of my knowledge, really aren't being trialed actively in human trials yet. Next slide. The question is, if you create this perfect copy of a gene, how do you get it into human cells in the place you want to take it? And so what scientists have been experimenting with for the past several decades is using experiments of nature. Basically, viruses are built knowing how to get into human cells. I mean, that's how we, those of us who have cold sores that appear whenever we get ill and stressed, have that happen. It's because we're storing that virus within our own cells, usually our sensory ganglia or sensory neuron cells. And so this is something that I don't believe that we have the technology yet to imitate, but viruses know how to do this. There are other ways, if you have a very simple transfer, where liposomes, which are just fat-laden membranes, can be wrapped around something to enter a cell. They're talking about nanoparticles, which would be a synthetic material, not fatty membranes, that might eventually be able to be used. And then there is some experimentation going on, trying to just take the naked DNA and see if it can't be introduced in a manner that won't get rejected. But most of the active trials now are using viral vectors. And the balance has to be to find a virus that would be able to get the synthetic gene material into the human body in the kind of cell or the location that you want without causing unwanted mischief. New slide. This just is a cartoon that's meant to give you a sense of this. So if you had that little green bauble, which is meant to represent a virus with its capsule, or what they call a capsid, with the triple helix of the DNA inside. What they're showing is that basically people take the newly synthesized gene, which would be the pink part, and put it in there after taking out the active infective parts of the virus, and then repackage it into this capsid, and then deliver either systemically, as people say, meaning to your whole system or whole body, usually through an intravenous infusion, or through an injection, for example, locally into the fluid that is in the eyeball and bathes the retina, or, as we will show you in a few slides, into the fluid that bathes the spinal cord so that the material can get close to the cells in the nervous system. Either systemically or locally, these viruses are produced. You see the virus going into what looks like a sphere or a ball, but which is really a cartoon of a cell, and then going through, and there's mechanisms that you probably remember from biology having to do with endoplasmic reticulum and ribosomes that then bring that material all the way to the nucleus, or that inner circle where the genetic material is. But this is the, on the slide you can see people mentioning the important parts, delivering that functional human gene into the cells you want in a manner where they will then activate and produce the kind of protein that's needed with enough efficiency, rapidity, and prolonged effect to take over the function that hasn't been working. New slide. This is just an example. Some of you might have heard of spinal muscular atrophy being one of the recent human trials that have led to commercial approval and commercial use. So we now at our institution are, when we say commercial, it means by prescription, by selecting out the appropriate patients, no longer as part of a trial, but just going ahead and doing it as therapy. On Asemdegene Parvovec, which is the long chemical generic name for what people call Zolgensma, is what is pictured here in this cartoon. And what you have at the ends of the synthetic virus are what they call ITR, or inverted terminal repeats. And essentially those are kind of placeholders to keep it in place. The promoter is something that will guide all of this to a particular type of cell. So some of the promoters will take it to all cells, others take it to nervous system cells, others primarily to muscle cells, and so forth. And then the transgene is the synthesized gene. So all of that is packaged inside that little capsule. So that would be a viral capsid or a viral genome. And many of those are used in one treatment. So an infusion over minutes or hours that might give a person something like 10 to the 12th power or 10 to the 14th power. So we're talking upwards of trillions of copies of these genes. Next slide. This slide is too complex to really absorb, but I wanted to make the point that as the scientists create this and make recommendations, it's being done with particular factors involved. There's sometimes some types of virus that will actually incorporate themselves into the human genes. That is beneficial because then if a cell did divide, it would reproduce that viral or newly introduced gene as well. So you would know that that gene would live on in the organism forever. On the other hand, there is potential for the control part of genes to be affected. And that's when they say there is potential for development of cancer, it is believed that one of those mechanisms that causes cancer in organisms is loss of control over the genetic material. So that's sort of the balance that you create. Now, for those reasons, that type of viral vector is not the one that's primarily being considered at this time for treatment of human genetic disease. We're using the non-integrating types, and in particular, the one that has most promise at the moment are adeno-associated viruses. They're not common, so most people don't have antibodies against them. And the diseases that they cause, even if people were somehow to get any of their own innate viral material, are not very serious. But the problem is that they're very small capacity. So there are some genes, like myotubular myopathy gene or spinal muscular atrophy gene, that work very well in an adeno-associated virus. But other genes, like Duchenne muscular dystrophy, is so big that the entire thing will not fit into one adeno-associated viral capsid. And so therefore they've had to make micro and mini dystrophins that contain part of the genetic material, but not all of it. Again, this is just a glimpse or a hint of a lot of the real thought that goes into creation of these types of viral treatments. Next. I wanted to give people a very, very quick sense of when we say intrathecal, what that means. Basically, the brain is there, above the ear, and underneath the beautiful hair of this child is the, brain that contains all of our neurons and really commands most of what happens in our body. That connects directly to the spinal cord. But as you can kind of see here, the spinal cord does not go all the way down to your tailbone. It stops just below your shoulder blade. And below that, there are nerve roots. In this rectangle that's blown up, you can see a stack of what looks like bone with marshmallows between, and that is the vertebra and the disc. That's in the anterior part. And then more posterior is the sac of fluid that bathes the spinal roots and spinal cord. And then behind that are the spinous processes. There's actually a ring of bone that goes from those tiny little bumps in the very back of your back around the side of the spinal cord to anchor onto those vertebrae. And so to deliver fluid or this genetic material for treatment to a place that's going to be right near the nerve cells, the lumbar puncture is used. And basically a very thin needle is used, as you can see in the lower part of the picture, and put just through the skin, just through the tissue underneath the skin, and just through the edge of that sac. That can take fluid out for analysis and put the medication in. Next slide. That turns out to be a very safe thing to do. Now, so we've talked a little bit about gene replacement therapy, but one of the things that we haven't talked about is cell-based gene therapy. And you can see on the left, they're talking about ex vivo gene therapy, where a gene is transferred in whatever way into the cells in a patient. And we've talked a little bit about ways to do that. But there is another way to try to get the gene into the human cell, and that is to remove the cell from the body. For example, taking a certain amount of blood with the blood cells out just by venipuncture, just like it would happen for a blood test. But this time, the cells are taken out very carefully and taken to a laboratory where scientists then do a gene modification in the laboratory, specifically in the way they want and need to those cells, and then transfer the patient's own cells back into their body. And that's frequently called, cell therapy, or some people have heard of CAR T. It's being used with lymphoma and leukemia, particularly when the cells that have the abnormality are blood cells. New slide. There are a lot of challenges that we have to face here. All genetic disorders don't have gene therapies available because the science has not been worked out, or perhaps because there are problems as scientists have been fighting with for the past 3 decades with Duchenne muscular dystrophy, where they know what the gene is and what most of the mutations are, but the gene itself is just too large to replicate. This whole process is very complex and very costly to develop. So it's not something that would lead itself easily to being done in rural areas or in all areas, irrespective of the amount of medical infrastructure and backup that exists. When we use viruses to be the transport agent to get the gene into the human body, if a person has already been exposed to that virus and has immunity, then that therapy cannot be used for that individual, at least for right now, until people come up with ways perhaps of dealing with that. If the target cell is replicating or dividing, so for example, muscle cells do divide, and people as they grow and have more muscle cells. We talk about teenage boys and teenage girls building muscle. They truly are adding on new cells. As that happens, the DNA from the new gene would be lost to the newly built cell unless it's incorporated into the host DNA. And then, as we talked about, there are concerns about incorporating your new gene into human genes because of a potential loss of control. New paragraph, or new, yeah, here we go. All of this has been changing so rapidly, it's very, very hard to keep up with. I put this slide together, and someone reminded me that this estimate of 400 active and recruiting clinical trials for gene and cell therapy applies only to the count for the United States. And if we step back a little bit and have a broader view and look throughout the world, the number is much larger. In fact, this website noted down here for the American Society of Gene and Cell Therapy lists a lot of the trials that exist. New slide. On the left side is just a list of therapies, gene-based therapies that have already been approved, and that, as I mentioned, can be prescribed without it having to be part of a clinical trial. Although, again, they are complicated and delicate therapies. My experience has been with SMA, but again, lymphomas, melanomas, a certain kind of breakdown in the retina, which is the nerve cell layer of the eye that gives you vision. So this therapy is something that restored vision. SCID is something that is a combined immunodeficiency because of a genetic abnormality. And this would reverse that. It's important to know that we should be thinking about these gene and cell therapies as something that can reverse and improve and stabilize a different kind of disorders, but we should not be thinking about them as a permanent cure because this is so new. We really don't have the experience and the observation period to really have determined whether all of these impacts that have seemed so positive and beneficial over the short term will be long lasting. And I think that's one of the questions that we need to ask, along with what strategies might we develop to repeat therapy or allow improvement in the duration of the therapies. And you can see on the right that there's a whole very, very long list of important disorders affecting children and adults that are in clinical trial at this time. And again, websites at the bottom that can give you more information about all of these. Next slide. So I hope that this makes it a little bit easier, but I, from having studied the whole area, realized that this is a very complicated area. It's one that all of us will be having to study and become more aware of as we work through our own health issues and those within our family and communities. Let me stop here and turn it back over to Erin and Marie to see what insight they can give people.
Sure. So thank you, Dr. Kuntz. It is a very complicated area, and you've made it less complicated for us, so we really appreciate the interesting overview that you provided. I'd now like to introduce Erin Ward, President, and Marie Wood, Vice President, MTM-CNM Family Connection, which is a nonprofit charitable organization with a mission to connect families affected by myotubular myopathy and/or centronuclear myopathy to resources, research, and relationships within this community. Both Ms. Ward and Ms. Wood are moms of children with MTM-CNM. I will turn it over to the both of you now to hear your stories.
Thank you so much, Katie. This is Erin speaking, and we just also wanted to thank American Brain Coalition, ASGCT, and Dr. Kuntz for collaborating on this webinar today. As Katie mentioned, we work within the MTM and CNM community, and our goal is to connect patients and families to research, resources, and relationships. And today we were invited to speak to gene therapy from the perspective of a patient community that is going through gene therapy and clinical trials. Specifically, we're going to focus on the process of our organization's involvement through different stages of clinical trials, as well as patient engagement and advocacy. In the photo next to my own photo, you'll see my family, my husband Mark, who is also on our board of directors, and my son Will, who is 18, living with X-linked myotubular myopathy. Marie, would you like to introduce your family?
Yes, thank you, Erin. So in the picture next to mine, you see my family. In the back row, you see my husband David. He's the tallest one. Next to him is Lucian, one of our son, and another son Blaze, who's holding our baby Kepler, next to my parents. And then I'm in the front there with Reed on my left and Paley on my right. And both Reed and Paley have X-linked myotubular myopathy. Clearly life has many challenges, but they are happy, wonderful kids, and they are the reason why we fight so hard to bring about betterment, not only in their lives, but in the wider community, which brings us to the next slide. So as Erin mentioned, the goal of our organization is to connect families to resources, research, and relationships, to really build and strengthen what we see as life-enhancing connections for the benefit of patients and families affected by MTM and CNM. And as the connections unite our community, we also create an effective community voice for advocacy. And it's this unity that also facilitates the spreading of knowledge, whether it's regarding standards of care, or sharing opportunities to be involved in studies, trials, legislative advocacy events, and so forth. So here you can see the latest photo from one of our biennial family conferences. That's one of our major projects. This is celebrating the 10th year of doing conferences. So up in the front, we have mostly kids and parents and extended family of siblings, grandparents, nurses, and then we also have medical research teams and representatives from pharma in the picture as well. So the conference itself is really a snapshot of all the different players in the world of our rare disease. So please don't be misled by the name Family Conference, because this is really a unique and great platform for all the stakeholders to be educated and empowered. And we'll be going into more detail as we go along about how the conference sets the stage and empowers gene and cell therapies. So Erin, in the next slide, can explain this with regards to preclinical trial.
We're very fortunate to be one of the communities where gene therapy trials are currently happening. It's actually our first clinical trial ever, so that's also exciting as well. What we found is through our conferences, we had a great opportunity to not only educate and connect families to ongoing studies and trials, but also we had a really unique opportunity to work closely with some researchers prior to the initiation of the gene therapy trial to do some data collection on site. So prior to the conference starting a few days before, we would have visiting clinical teams who were really looking at how to best come up with ways to measure possible outcomes that we may see after treatment. And then specifically, we work with a team who was collecting data on breathing measurements. As you might have seen from the photos, our rare neuromuscular condition, X-linked MTM, greatly impacts respiratory function, and many of our individuals live with the assistance of tracheostomies and ventilators. And also their muscle function and movement overall is greatly profoundly impacted, and many also use the aid of wheelchairs. So measuring improvements was something that they were specifically looking to see. If someone was treated, what would a potential outcome look like, and how would that be measured? So we work with a team focusing on respiratory data collection as well as movement collection. So this was such a unique opportunity for researchers to very quickly have interaction with patients that have X-linked MTM, and also over a period of a few days collect data that potentially would have taken months or years to collect in other settings. Also, the conversation of natural history studies is an important one. Bringing the researchers together on site. Back when we started in 2009, there was discussions about how researchers had worked together virtually over the years, but had never even met each other in person. And coming together in person over a short period of times, along with families, stems from creativity and discussion, and also you see the value of what you're working for when you see the patients right in front of you. Generates the initiation of natural history studies, accelerates publication, and so forth. Also, we've had very meaningful conversations over the years with researchers engaging in this work, discussions even over lunch and in casual settings throughout the weekend about what's most meaningful to patients, what kind of endpoints and biomarkers might be incorporated into a clinical trial. So we want to include this specifically for those of you that may not yet be in a trial setting, thinking about ways you can organize yourself, bring your community together, and think about this type of collaboration. Next slide for Marie.
Right, and so as a patient advocacy group, engaging with researchers and industry, our conferences really are an ideal place for updates on research into possible treatments, such as gene therapy. Just to give you a kind of an overview, 10 years ago at the first conference, which was in 2009, we learned about studies of the disease in mice models and zebrafish. Then in 2011, we were hearing about progress in those models, as well as success in gene therapy in large animal trials. By 2013, industry partners were attending and sharing at our conference about the types of therapies they were pursuing to bring to market. And then in 2015, we were discussing enrollment into human preclinical trials. And in 2017, we were informed that the first dosing in the human clinical trial would be happening shortly thereafter. In 2019, at the latest conference, we heard about the success in trials and that there could possibly be licensing of the product within the next year. In 2020, we hope that potentially there may be patients receiving the drug at that time. So we're cautiously optimistic about that. And we really, as Erin pointed out, provide opportunities for recruitment into the studies, such as the natural history studies and trials. We provide forums for discussing areas of future research. As we listen to the community, we again strengthen the amplitude and power of that community voice, addressing the needs of today as we work towards that brighter future. We provide outreach to the community on behalf of industry for recruitment into focus groups and advisory boards, in which the hopes and needs of patients and caregivers are incorporated into meaningful goals of the therapies that are being funded and pursued. So when I attended one such parent advisory board, virtually, it was virtually unanimous among the parents in that room that they had a desire for their child to be strong enough to breathe independently of the ventilator. So that's a meaningful goal to us, and it's important for industry to know those things when they're developing treatments. And as our conference only takes place every 2 years, the patient advocacy leaders continue the collaborative discussions at other meetings and through phone calls and such. So we really keep the discussion going. Next slide.
So we first thought we'd talk about the benefits of gene therapy for our community, and we were very hopeful about the preliminary data. Also, I think it's an understatement to say that if effective, the benefit could be profound for our families and our loved ones living with this disease. Currently, it's important to also note that although there has been positive outcomes in preliminary data, there is no currently approved treatment for MTM. So even in my son's lifetime, 18 years ago, the pace that this has gone has been quite extraordinary. We were still learning about the disease 18 years ago. There were no therapeutic interventions on the horizon. And we are actually very fortunate that we have dedicated researchers, that we actually have several different strategies being looked at, with gene therapy being the first one in active clinical trials. So part of our role is, we'll talk a little bit more in a bit about educating the community, but also, as promising as the outcomes have been through the data shared publicly, it's also important to remain cautiously optimistic and to understand that also the idea of gene therapy may be embraced differently across our community. It was very interesting at our conference this summer. A family member stood up and spoke about how his son actually would potentially not choose to have the therapy, or also the idea of how important it is to continue to think about caring for your child in the daily presence of what they need and making decisions in the here and now, where the possibility of gene therapy is not right now on the market. And going into that is a little bit talking more about the challenges. So Marie is going to speak more to that in the next slide.
Right. So time is probably one of the biggest issues on everyone's mind when they think about a therapy that's on the horizon. It's a huge issue. Sadly, tubular myopathy has a high mortality rate, especially within the first few years, and most patients with MTM do not reach adulthood. And even though the disease is not technically degenerative, as a child grows older, more issues arise and their care becomes increasingly complex. So even if a therapy can make muscles stronger, it may not be able to undo some of the complexities of the disease's progression through aging. So there's really this profound sense of urgency that's pervading the community. Another challenge is eligibility. So the picture you see over on the right is of some of the kids at our 2017 conference in Nashville, Tennessee. Based on age factor alone, most of those children would not have been eligible to participate in a trial. So questions like who can participate in trials and who will receive the drug can really be very emotional. And not everybody handles it well when they find out that their child, and understandably so, when they find out that their child may not be eligible for an investigational new drug that could drastically improve their quality of life and survivability. So we are very sensitive to those needs, especially having older children ourselves. And our goal as patient advocates is to remind everyone that not participating in a trial does not mean that a patient never receives the drug, and that eligibility for a trial has more to do with designing for maximal success of a trial, and it's not necessarily limiting who can ultimately receive a treatment. As patient advocates, we want to see successful trials that allow for the drugs to reach all of those who are affected by the condition, whether young or old, mildly or severely affected. No man, woman, or child left behind. So sometimes that means asking hard questions and putting pressure on those who hold power over those choices, and that's something we've always been willing to fight for. So as Dr. Kuntz mentioned, the AAV immunity issue, so that is another challenge. With the viral vectors for gene therapy, you have to deal with the possibility that a particular subject could be immune. Now, immunity is tested prior to the administration of the drug and the trials, but it would be a decisive limiting factor in receiving the therapy. So that can be a concern for the community. Another emotional topic deals with this idea of expanded access or compassionate use. And that's when, that's the case when a trial drug can be given to a non-trial participant who is in a serious or life-threatening condition, but is not eligible to participate in the trial. So in our community, most of the patients are children, which itself complicates and increases the emotions around that issue. Furthermore, you could rightfully say that every child with MTM is in a serious life-threatening condition just based on the nature of the disease. And especially for rare disorders like MTM, there's a desire and critical need to collect data in the trials, which expanded access could complicate. So if patients outside the trial receive a non-approved drug, there could also be side effects and other undesirable outcomes due to the effects of the drug not being fully understood, and you can't undo the injection of a gene therapy. Oops, sorry. So, and then balancing the hope versus the hype is really this idea that, you know, there are all these positives and benefits, as Erin talked about, but there are still these challenges. So we really want to encourage families to make decisions that will allow their child to best thrive in the present moment and, you know, not put their lives on hold waiting for the cure. We don't want to present an unrealistic pictures that all the kids will be running around as if MTM had never been a part of their lives, but we do want to spread the word of hope and maintain our optimism about what the future holds if we're willing to work for it. And that's where education is really important too. So that brings us to the next slide.
Thanks, Marie. So a big part of our organization is promoting education, and we do that at our conferences as well as through online social media and other materials that we develop. And we would also like to, again, thank American Society of Gene and Cell Therapy. A little while ago, Allison from ASGCT had invited us to participate in collective phone calls, specifically developing some educational materials on Gene Therapy 101 and specifically on MTM. And we did those phone calls along with our colleagues from the Myotubular Trust, the UK charity for myotubular myopathy, as well as members from the MDA. And I really encourage you to visit the website and look at those materials. They're very informative, and we distributed them as well as our conferences. The image here is one that our team had put together prior to that project, especially when our trials first started, to help inform the community that this was actually a combined phase 1 and phase 2 clinical trial, and to try to demonstrate the length and the process that the drug development has to go through. And of course, now we're working specifically with biologics approval process versus drug approval process with looking at gene therapy. A next part of our efforts for patient engagement is an advocacy, and Marie will speak with that next.
Right, so as these new gene therapies for various diseases come into play, there's this ever-increasing role for patient advocates. So recently we worked alongside others in the community to present a case to the CDC for a unique ICD code. So that's the International Classification of Diseases, a unique code for X-linked myotubular myopathy. And this type of code is important. It can facilitate medical billing, also diagnosis, treatment, outcomes, tracking by government agencies that can give us more data on mortality and other issues arising, and even international statistics, which can really add to the understanding of a disease. Since these new gene therapies typically involve one dose and are potentially curative, patient advocates can become involved in legislative advocacy and encourage legislation pertaining to new payment models that are centered on value-based care and payments over time, so that the financial returns are worth the investment that the company is making. I mean, this is important to us as a patient advocacy organization because we want these companies to stay in business and keep producing the drug for future generations of patients. So we've collaborated with and connected our community to organizations like the Every Life Foundation, which seeks to influence bipartisan legislation and increase funding of the FDA and NIH so that we can support the development and access of treatments to those who need it. Rare Disease Week on Capitol Hill. This is a photo taken from that with me and 2 other amazing parent advocates in our community. So Rare Disease Week on Capitol Hill and Rare Across America are some of Every Life's programs, and they set up meetings between representatives and senators and the voters in their state or district who can represent the needs of those impacted by rare disease. At our recent family conference, we announced the creation within our organization of a Committee on Legislative Advocacy, or COLA, and we're working to establish a network of informed families affected by MTM and CNM in each state to keep them abreast of relevant legislation, connect them with opportunities to participate in events such as Rare Disease Week, to equip them with tips and tools for making the best of meetings with their members, and to, again, amplify our unified voice where it counts. So next slide.
And adding on to the advocacy piece, one of the best things that we've done as our organization is to get connected with other rare disease communities. And we've done so through events such as the Global Genes Patient Advocacy Summit, NORD meetings, and also state-based rare disease meetings. So all of this is about collaboration, especially in the climate of these new therapies such as gene therapy. We all need to be sharing resources and what has gone on. And we've specifically reached out to communities that are ahead of us in this process. And today we were very grateful to be a part of sharing some of the insights we've had so far in the journey for those that may be a little bit further back in development. So it's all about collaboration, and we invite you to explore these different avenues of advocacy and engagement. And next slide, we just have our ways that you can connect with us.
Excellent. Well, thank you, Erin and Marie. Parents like you remain the greatest advocates, and your organization is truly a good example of a community uniting to fight for this devastating disease. We have a couple minutes And I know you've answered a lot of my questions that I've written down, and I believe you've answered most questions as I'm looking through. I do have one question that came in. What type of physician refers a patient to gene therapy? Maybe Dr. Kuntz, you can answer that.
Well, again, it depends on at what stage the drug is at in development. I think that anybody who makes a diagnosis of any of these disorders, and so sometimes it will be a geneticist, sometimes it would be a specialty physician like an ophthalmologist for the eye diseases or a neurologist for the neuromuscular diseases. So anybody who makes the diagnosis, is undoubtedly reaching out to both make available the standard of care, the basics that have been in place for a period of time, as well as to offer the family the opportunity to be treated with some new treatments, either as part of a clinical trial, for which they need to be educated and give informed consent, knowing that it's still at the trial phase, or for some of these ones that have already been approved, then each of them is slightly different in terms of what skill set it takes for a person to administer those therapies. They're usually a bit more complicated than just writing a prescription and having someone go to the pharmacy and pick it up. So the ones I'm involved with, for example, we have a hospital-based team with nurses and therapists and physicians and pharmacists who are all involved in making sure it goes well. And so the people who refer to us are the people who make the diagnosis.
Great. Thank you. And actually, speaking of clinical trials, the site that you had provided, the AFGCT clinical trials site, I will share that on the ABC's website and follow up with participants as well, because I think that would be very helpful for folks. Well, it looks like we are almost at the top of the hour. So I would like to thank once again the American Society of Gene and Cell Therapy and MTM-CNM Family Connection for co-sponsoring today's educational webinar on gene therapy. This webinar was recorded and it'll be available on the ABC's website. But I'd especially like to thank Dr. Nancy Kuntz and Erin Ward and Marie Wood. Thank you also, all of you, for participating. The ABC's program committee welcomes your input as we brainstorm future ABC educational webinars such as these, and please contact any of the ABC staff with any ideas you may have. Thank you all and have a great day.